Welcome to the deep dive. Now, when you think about the human body, you probably picture muscles, bones, organs, right? The visible stuff.
Yeah, the structures.
Exactly. But beneath all that, there's this invisible world, a constant chemical conversation happening. And to really get how you move, think, breathe, well, you have to understand the language it's speaking, chemistry. Now, I know what you might be thinking. Chemistry sounds a bit intimidating, maybe dry, lots of formulas.
It can seem that way sometimes.
But our mission today is to really distill the essential chemical concepts from, a typical anatomy and physiology foundation like you'd find in a core textbook.
Yeah.
We want to make it clear, accessible.
Right. We're not just memorizing terms.
No, we're digging into the why things work the way they do. From a simple blink to your heartbeat, it all starts here.
Absolutely. Think of this as like the shortcut to getting the fundamentals. It's amazing, really, when you realize everything. Every cell doing its job, every organ system, it's all driven by chemical interactions.
Everything.
Pretty much everything. From the tiniest atoms, the smallest particles, all the way up to those big, complex molecules that, well, make you you. No chemistry, no life. Simple as that.
Okay, let's unpack that. Why start an A&P deep dive with chemistry? Seems a bit backward if you want to get straight to, say, muscles and nerves.
It might feel that way, but your body isn't just parts. It's basically a giant, incredibly organized chemical factory.
A factory?
Yeah, running thousands, maybe millions of chemical reactions constantly, right now at speeds that are just, well, mind-boggling.
Oh, yeah.
And these reactions, they don't just help the body work, they are the work.
They are the processes.
Exactly. You take a step. That's chemistry. Digesting food, pure chemistry. Even the thought you're having right now.
Listening to us talk about chemistry.
That's chemical and electrical signals firing. So you see, if you understand the chemistry, you grasp the core mechanics of life itself.
Okay, makes sense. So where do we begin? The real basics.
Let's start with matter and energy, the fundamental duo.
Right. Matter. Pretty straightforward. It's anything that takes up space and has mass. Your bones, blood, the air you just inhaled. All matter. And just a quick point, mass isn't quite the same as weight, is it? Mass is constant. Weight depends on gravity. Like you weigh less on a mountaintop.
Good distinction. Your mass stays the same, gravity's pull changes.
Then there's energy, the capacity to do work, to put matter in motion. You can't always see it, but you see its effects, right?
Definitely. Like a baseball player hitting a home run versus just bunting. Big difference in energy used.
Exactly.
And energy comes in two main flavors, you could say. Kinetic and potential.
Kinetic. That's energy in action.
Right. A bouncing ball, moving blood cells, even atoms are always vibrating. That's kinetic energy. It's energy doing something.
Okay. And potential.
That's stored energy, energy that could do work. Think of water behind a dam, or the chemicals in a battery.
Or even just sitting still. My leg muscles have potential energy.
Precisely. They have the potential to contract. And when that potential energy is released, bang, it becomes kinetic energy, action.
Okay, this is where it gets really interesting for the body. There are 4 key forms of energy we deal with. First, chemical energy.
The big one.
Yeah, stored in the bonds between atoms in molecules, like the energy in your food.
Yeah.
Your body converts that into kinetic energy to move your arm, for instance.
Okay. And crucially, it captures that energy in a molecule called ATP.
Ah, yes, ATP, adenosine triphosphate. We'll definitely come back to that.
Yeah.
That's like the body's ready to spend cash, right?
Exactly. The direct energy currency for cells.
Okay, second form, electrical energy. Movement of charged particles.
Think nerves.
Right. Nerve impulses are basically tiny electrical currents, and the heart's rhythm, also electrical.
Which is why a nasty electric shock is so dangerous. It messes with those internal signals.
Makes sense. Third, mechanical energy. This is more direct, moving matter.
Like riding a bike. Muscles pulling on bones.
Yep. And fourth, radiant energy. Electromagnetic radiation. Travels in waves. Visible light lets us see. UV waves from the sun help us make vitamin D.
But can also give you a sunburn.
Right. A double-edged sword there.
So if we connect this back, a key thing about energy is conversion. You change it from one form to another. Chemical to mechanical, for example.
But it's not perfectly efficient, is it?
Never. Some energy is always lost as heat. It's not truly lost, just becomes less useful for work. Think of an engine getting hot.
So when my muscles contract, they generate heat.
They do. Every energy conversion in your body releases heat, and that's actually vital. It helps maintain your core body temperature.
Which keeps all those chemical reactions running at the right speed.
Exactly. Body temperature is crucial for reaction rates. Too cold, things slow down. Too hot. Well, we'll get to that.
Okay, so we have matter and energy. What is the actual matter made of?
Yeah.
Down to the basics. Atoms and elements.
The building blocks.
Elements are unique substances like oxygen, carbon, hydrogen, nitrogen. Can't break them down further by normal chemistry. And those four, they make up, what, 96% of our body weight?
About that, yeah, the vast majority. Though other trace elements are still essential.
And each element is composed of atoms, tiny particles. Remember, atom means indivisible, but we know now they can be split, right?
We do, into subatomic particles. But an atom is sort of the smallest unit that still retains the element's properties.
Okay, so what's inside an atom?
Right, let's picture it. You've got a central nucleus, really dense, packed with positively charged protons.
Positive charge. Got it.
And neutrons, which have no charge. They're neutral. These give the atom almost all its mass.
Protons and neutrons in the nucleus. Heavy stuff.
Then, orbiting way out from the nucleus are the electrons. Tiny, negatively charged particles. Practically massless compared to protons and neutrons.
Negative charge orbiting outside.
And the key thing, atoms are electrically neutral. The number of positive protons equals the number of negative electrons. Balances out.
So it's balanced. But you said orbiting way out. How far?
The scale is amazing. If a hydrogen atom, the simplest one, was scaled up to the size of a football stadium, the nucleus, that one proton, would be like a single gumdrop sitting on the 50-yard line.
Wow.
And the electron. It would be like a fly buzzing around somewhere inside that huge stadium. Atoms are mostly empty space.
That's hard to picture.
Oh.
Mostly empty.
Yeah.
Okay, so how do we tell atoms apart? Identify them.
Key identifiers. First, the atomic number. That's simply the number of protons. It defines the element. Hydrogen always has one proton, helium always two.
Atomic number protons equals element identity. And since atoms are neutral...
It also equals the number of electrons.
Right. Then there's mass number.
That's protons plus neutrons. Tells you about the atom's mass, its weight, essentially.
Okay, and atomic weight is slightly different, an average.
Yeah, it's the average weight of all the isotopes of an element. Don't worry too much about that distinction for now.
Isotopes. You mentioned those. What are they again?
Good question. Isotopes are atoms of the same element, so they have the same number of protons.
But different numbers of neutrons.
Exactly. So they have different mass numbers, like hydrogen usually has no neutrons, but deuterium has one, tritium has two, same element, different weight.
Got it. And some isotopes are unstable.
Yes, those are radioisotopes. Their nuclei are unstable and they decay, spitting out radiation alpha, beta, or gamma particles.
Radiation sounds bad.
It can be. High doses damage tissues. Think radon gas causing lung cancer. But this decay happens at a predictable rate.
Ah, so we can use them.
Precisely. We use them in medicine all the time. Iodine-131 to check thyroid function. PET scans use radioactive tracers. Cobalt-60 targets cancer cells. Invaluable diagnostic and treatment tools.
So useful, but potentially dangerous.
Okay.
From atoms, we build up molecules and mixtures.
Right. Next level of organization.
A molecule is just two or more atoms bonded together, right? Like O2, oxygen gas.
Yep, that's a molecule of an element.
But if the atoms are different types, like hydrogen and oxygen making water, H2O, or carbon and hydrogen making methane, CH4, that's a compound.
Correct. A compound involves two or more different kinds of atoms bonded together.
And compounds can have totally different properties than the elements that make them up. You mentioned sodium and chlorine.
Classic example. Sodium is an explosive metal. Chlorine is a poisonous gas. Bond them together. You get harmless table salt, NaCl. Chemistry is transformative.
Amazing. Okay, so that's molecules and compounds, chemically bonded. What about mixtures?
Mixtures are different. Components are just physically mixed, not chemically bonded.
Like tossing nuts and bolts together in a jar.
Sort of, yeah. And most stuff in your body is actually in mixtures. Three main types to know.
Okay. First.
Solutions. These are homogeneous, meaning uniform throughout. Think salt water or sugar dissolved in water. The solute particles are tiny, invisible, and they don't settle out.
Like saline solution used in hospitals.
Exactly. We talk about concentration here. Percent solution, milligrams per deciliter, like for blood sugar, or molarity for precise chemical work.
Molarity involves Avogadro's number, right? For counting atoms.
That's the one. Very precise for body fluids.
Okay. Solutions are uniform. Next.
Colloids or emulsions. These are heterogeneous, often look milky or translucent. Think Jell-O before it sets or milk.
Or inside our cells, cytosol.
Perfect example. Cytosol, blood plasma. The solute particles are bigger than in solutions, large enough to scatter light. That's why they look cloudy, but they still don't settle out.
And they can change consistency, like liquid to
Yes,
gel.
Sol-gel transformations.
No.
Really important inside cells.
Okay. And the third type?
Suspensions. Also heterogeneous, but the particles are much larger, often visible, and crucially, they will settle out over time.
Like sand in water. Or blood.
Blood is a great example. Let it sit, and the red blood cells settle to the bottom. It's a suspension.
Right. So solutions, colloids, suspensions. How do we really tell a mixture apart from a compound then? What's the key difference?
This raises an important question. How do we tell these apart? Well, the biggest thing, no chemical bonds form between components in a mixture.
So you keep their original properties. Salt is still salty in water.
Exactly. You can usually separate mixtures by physical means, filter the sand, evaporate the water. Compounds. You need chemical reactions to break those bonds.
And compounds are always homogeneous, same composition throughout. Mixtures can be either homogeneous solutions or heterogeneous colloid suspensions.
You got it. That distinction is fundamental.
Okay, so let's talk about the glue itself.
Yeah.
Chemical bonds. What holds atoms together in molecules and compounds?
It all comes down to electrons. Specifically, the electrons in the outermost energy shell, the valence electrons.
Valence shell, the outermost one.
Right. Atoms, well, they want to be stable, chemically inert. And for most, stability means having a full valence shell, usually 8 electrons.
The octet rule, aiming for 8.
Exactly. Or 2 for the very first shell, like helium. The noble gases, helium, neon, argon, already have full outer shells. That's why they're so unreactive or inert. They're already happy.
Okay, so atoms that don't have a full shell need to interact to achieve that stability. How?
3 main ways they form bonds. First, ionic bonds.
Ions, charged atoms.
Right. In ionic bonding, one atom actually transfers 1 or more electrons completely to another atom.
Gives them away.
Yep. The atom that gains electrons becomes negatively charged an anion. The atom that loses electrons becomes positively charged a cation.
And opposites attract.
So the positive cation and negative anion are strongly attracted to each other. That electrical attraction is the ionic bond. Think sodium chloride, NaCl again. Sodium gives an electron to chlorine. Na plus attracts Cl. Boom. Salt crystal.
Transfer creates ions, ions attract. Got it. Second type?
Covalent bonds. Here, instead of transferring, atoms share electrons.
Sharing is caring for atoms.
Something like that. By sharing pairs of electrons, atoms can effectively fill their valence shells. They can share 1 pair, single bond, 2 pairs, double bond, or even 3 pairs, triple bond.
Like in hydrogen gas, H2, oxygen gas, O2, nitrogen gas.
Perfect examples. Single, double, triple bonds, respectively. Methane, CH4, is another good one with single bonds.
Now, is the sharing always equal?
Ah, good point. No. If electrons are shared equally, the resulting molecule is nonpolar, like carbon dioxide, CO2. It's symmetrical, so the electron pull cancels out.
Okay, equal sharing, nonpolar.
But if one atom attracts the shared electrons more strongly than the other, we call that being more electronegative. The sharing is unequal. This creates a polar molecule or a dipole.
Like water, H2O.
The classic example. Oxygen is much more electronegative than hydrogen. It pulls the shared electrons closer, plus water has that bent V shape.
So the oxygen end is slightly negative, and the hydrogen ends are slightly positive.
Exactly. That polarity is hugely important for water's properties, as we'll see.
Okay, ionic is transfer, covalent is sharing, polar or nonpolar. What's the third type?
Hydrogen bonds. Now, these aren't true bonds like ionic or covalent where electrons are transferred or shared between the bonded atoms. They're more like strong attractions.
Attractions between molecules.
Usually, yes, or between different parts of a very large molecule. They form when a hydrogen atom, already covalently bonded to a very electronegative atom like oxygen or nitrogen,
Is attracted to another nearby electronegative atom.
precisely. They're weaker than covalent or ionic bonds, but there are often tons of them.
What do they do?
So much. They're responsible for water's surface tension, why water beads up, why insects can walk on it.
Okay.
And absolutely critical inside large biological molecules like proteins and DNA. They help maintain the specific 3D shapes of these molecules.
That shape determines function, right?
You got it. Without hydrogen bonds, proteins wouldn't fold correctly, DNA wouldn't hold its double helix, life would unravel.
Wow. Okay, weak bonds, but massively important. So we have atoms, molecules, bonds. Now, how do they change? Chemical reactions.
The action. This is where bonds are formed, broken, or rearranged. Everything happening in you right now involves chemical reactions.
And we represent them with chemical equations, right? Reactants on one side, products on the other.
Yep. Reactants, products. Shows what goes in and what comes out.
What are the main types of reactions?
Generally, we classify them into three major patterns. First, synthesis reactions, building things up.
Anabolic, A plus B, EB.
Exactly. Like amino acids joining to make a protein. Constructive.
Second, decomposition reactions, breaking things down.
Catabolic, A plus B. Like glycogen breaking down into glucose molecules. Destructive.
And there's a special type of decomposition, redox.
Ah, yes. Oxidation reduction reactions, or redox. These are super important. They involve the exchange of electrons. One substance is oxidized, loses electrons. Another is reduced, gains electrons.
Oil rig oxidation is loss. Reduction is gain.
That's the mnemonic. This is how your body gets energy from food. Glucose is oxidized, oxygen is reduced, and energy is released to make ATP.
Okay. Synthesis, decomposition, including redox. Third type,
Exchange reactions or displacement reactions. Basically, parts of molecules swap partners. AB plus C, AC plus B.
like musical chairs for atoms.
Kind of. A key example is ATP transferring its phosphate group to glucose. That's an exchange reaction. ATP plus glucose, ADP plus glucose phosphate.
Okay, got the types. What about energy flow and reactions?
Reactions either release or absorb energy. Exergonic reactions release energy. They're typically catabolic, like breaking down fuel. The energy is now available.
And endergonic reactions require energy input to happen. They absorb energy, usually anabolic, building complex molecules.
Right. And the body cleverly couples these. The energy released from exergonic reactions, like glucose breakdown, is captured, often in ATP.
And then that ATP energy is used to power the endergonic reactions, like protein synthesis.
Exactly. One hand washes the other, energetically speaking.
Are reactions always one-way streets?
Theoretically, most chemical reactions are reversible. A plus B, AB. The double arrow shows reversibility.
But you said earlier, many in bodies seem irreversible.
Right, because either the energy release is immediately used or one of the products is quickly removed or used up. Think about CO2 it's produced, but then you exhale it, so the reaction keeps going in one direction.
Ah, okay. What about chemical equilibrium?
That's when the rate of the forward reaction equals the rate of the reverse reaction. It doesn't mean the amounts of reactants and products are equal, just that there's no net change anymore. Things are still happening, but the conversions balance out.
Okay. So what does this all mean for us? What speeds up or slows down these vital reactions?
Several key factors influence reaction rates. Temperature is a big one. Higher temp, faster reactions, usually.
But too high is bad for us.
Very bad.
Like-
High fevers can start to damage proteins. We need that optimal body temperature.
What else?
Concentration. More reactants packed together means more collisions, faster reaction.
Makes sense. And particle size.
Smaller particles move faster and have more surface area, so they react faster than large chunks.
Okay. Temp, concentration, size.
Yeah.
Anything else?
Yes, catalysts. These are substances that dramatically increase reaction rates without being used up in the reaction themselves.
They just help it happen faster.
Exactly. And in our bodies, the catalysts are absolutely critical. They're called enzymes.
Biological catalysts. Mostly proteins, right?
Mostly proteins, yes. And they are essential for life. Without enzymes, reactions would be far too slow at our normal body temperature.
Okay. Enzymes are key. Let's shift gears slightly to the types of compounds in the body. We have inorganic and organic. Let's start with inorganic compounds.
Right. These generally lack carbon, though there are exceptions like CO2. But things like water, salts, acids, and bases are inorganic and absolutely vital.
And water is number one, right? Makes up most of our body weight.
Easily. 60–80% of most cells. And its properties are just incredible, mostly due to its polarity and hydrogen bonding.
What makes it so special?
Well, first, its high heat capacity. It absorbs and releases large amounts of heat without changing its own temperature much.
So it helps keep our body temperature stable, protects us from sudden changes.
Exactly. Buffers against external temp changes and heat generated by muscles. Second, its high heat of vaporization.
Meaning it takes a lot of heat to turn liquid water into gas.
Right, which makes sweating a super effective way to cool down. As sweat evaporates, it takes a lot of heat with it.
Clever. What else?
Its polar solvent properties. Water is called the universal solvent for a reason. Its polarity lets it dissolve salts, sugars, and other charged or polar molecules.
So it's the body's main transport medium. carrying nutrients, wastes, gases, and blood.
Absolutely. And it forms hydration layers around large molecules like proteins, keeping them dispersed, forming those biological colloids we talked about.
It's also reactive, isn't it?
Yeah.
Involved in breaking down molecules.
Yes, it's a key reactant in hydrolysis reactions. And finally, it provides cushioning. Think cerebrospinal fluid protecting the brain.
Water's amazing. Okay, what about salts?
Salts are ionic compounds like sodium chloride, potassium chloride, calcium phosphates. When they dissolve in water, they dissociate into ions.
Charged particles again. Electrolytes.
Exactly. And these electrolytes are crucial. Sodium Na+ and potassium K+ for nerve impulses and muscle contractions. Calcium Ck2+ for bones, teeth, muscle contraction, blood clotting. Iron F and hemoglobin for oxygen transport. The list goes on. They're essential minerals.
We need those electrolytes. Okay, last inorganic group, acids and bases.
Also, electrolytes. They dissociate in water, but they specifically affect the concentration of hydrogen ions, H+.
Right. Acids are proton donors. They release H+ ions.
Correct. Like hydrochloric acid, HCl in your stomach. More H+ means more acidic.
And bases are proton acceptors. They take up H+ ions.
Yes. Things like hydroxides like NaOH or bicarbonate ions, HCO3 in your blood. They reduce the H+ concentration, making things more alkaline or basic.
And we measure this with the pH scale.
Yeah.
0 to 14.
Yep. Logarithmic scale. 7 is neutral. Below 7 is acidic. More H+. Above 7 is alkaline or basic. Less H+.
Then our blood needs to stay in a very narrow range, right? Slightly alkaline.
Very narrow. About 7.35 to 7.45. Deviations can be fatal quickly.
So how does the body manage that? Acids and bases neutralize each other, right? Acid plus base water plus salt.
They do, but the body mainly relies on buffers.
Buffers. They resist pH changes.
Exactly. They're chemical systems that can bind H+ ions if pH drops, gets too acidic, or release H+ ions if pH rises, gets too alkaline. They act like sponges for H+I.
Like the carbonic acid-bicarbonate system in blood.
That's the major one. It works constantly, along with your lungs, breathing out CO2, which affects acidity, and kidneys, excreting acids bases, to maintain that critical pH balance. Homeostasis in action.
Incredible balancing act. Okay, that covers the key inorganics. Now for the big players, organic compound.
The molecules of life. Carbohydrates, lipids, proteins, and nucleic acids. They're unique to living systems and all contain carbon.
What's so special about carbon?
Carbon is amazing. It's electroneutral. It doesn't easily gain or lose electrons. It prefers to share them. And it has 4 valence electrons.
So it can form 4 covalent bonds.
Right. Which allows it to link up with other carbons to form long chains, branch chains, rings. Incredibly diverse and complex structures. It's the perfect backbone for large, intricate molecules.
The scaffolding of life.
You could say that. And these large organic molecules are often polymers built from smaller repeating units called monomers.
Like beads on a string.
Good analogy. And the body builds these polymers using dehydration synthesis.
Dehydration. Removing water.
Yes. A hydrogen atom, H, is removed from one monomer, and a hydroxyl group, OH, from another. They combine to form water, H2O, and the monomers link up where the water was removed.
Building by removing water, how does it break them down?
The reverse process. Hydrolysis. Hydro means water, lysis means splitting. You add a water molecule back to break the bond between monomers.
Building and breaking, using water. Fundamental processes.
Okay, let's look at the specific classes. First, carbohydrates.
Sugars and starches. Their monomers are monosaccharides or simple sugars.
Like glucose, the
Yeah.
famous blood sugar.
Exactly. Glucose is the main fuel source for our cells. Fructose, fruit sugar, and galactose are others.
What happens when you link two monosaccharides? You get a disaccharide, sucrose, table sugar, glucose plus fructose, lactose, milk sugar, glucose plus galactose, maltose, glucose plus glucose.
And linking many sugars.
That forms a polysaccharide. These are large, often insoluble molecules. Plants store glucose as starch. Animals, including us, store it as glycogen.
Glycogen, stored mainly in the liver and muscles.
That's right, ready to be broken down back into glucose when needed for energy.
So primary function of carbs. Quick, easily used energy source. Fuel for making ATP.
Primarily, yes, though they have some minor structural roles, too.
Okay, next class. Lipids, fats, oils, waxes.
Right, defined by being insoluble in water. They also contain carbon, hydrogen, and oxygen, but much less oxygen relative to hydrogen than carbs. Some contain phosphorus, too.
What are the main types?
First, triglycerides or neutral fats. These are what we typically call fats and oils. They're made of glycerol and 3 fatty acids.
Function.
Major function is energy storage, very efficient, more energy per gram than carbs, also insulation and protection, cushioning organs.
And we hear about saturated versus unsaturated fats.
Right. It refers to the bonds in the fatty acid chains. Saturated fats have only single bonds, their chains are straight, they pack tightly, and are usually solid at room temp, like butter. Unsaturated fats have one or more double bonds, causing kinks in the chains so they don't pack well, and are usually liquid oils, like olive oil.
And trans fats are bad, omega-3s are good.
Generally, yes. Trans fats are artificially saturated oils linked to heart disease. Omega-3s are essential unsaturated fats with health benefits.
Okay. Triglycerides for storage. What else?
Phospholipids. These are modified triglycerides. They have a glycerol backbone, two fatty acid tails, which are nonpolar, hate water.
But the third position has a phosphorus-containing group, which is polar, loves water.
Exactly. A polar head and nonpolar tails. This dual nature is perfect for forming cell membranes. They arrange themselves in a bilayer, tails inward, heads outward, creating a barrier.
Fundamental structure of cell membranes. Crucial. Okay, third lipid type.
Steroids. These have a completely different structure. 4 interlocking hydrocarbon rings. Flat molecules.
That's cholesterol.
Cholesterol is the most important steroid. It's made by our liver, and we get it from animal foods. Despite the bad press, it's essential.
Essential. How?
It's a structural component of cell membranes, keeps them fluid. It's the raw material for making vitamin D steroid hormones like testosterone, estrogen, cortisol, and bile salts needed for digestion. You can't live without it.
Good to know. Any other lipids?
Briefly, eicosanoids, derived from fatty acids, involved in things like blood clotting, blood pressure regulation, inflammation, labor contractions. Prostaglandins are a key example.
Okay. Carbs, lipids.
Yeah.
Now for the workhorses. Proteins.
Arguably the most complex and versatile, make up 10–30% of cell mass. Basic structural material, but they do almost everything else too.
Like enzymes, hemoglobin, muscle proteins?
Right. Their functions are incredibly varied, and they're polymers of amino acids.
There are 20 common types of amino acids.
Yep. They all have a common core structure, but differ in their side chain, or R group. That R group gives each amino acid its unique properties.
And they link together via peptide bonds.
Correct. Formed by dehydration synthesis between the acid group of one amino acid and the amine group of another. A chain of amino acids is a polypeptide.
And the sequence of amino acids is key.
Absolutely critical. That sequence is the primary structure. It dictates everything else.
What are the other levels of structure?
The primary chain coils or folds into secondary structures, mainly the spring-like alpha helix or the folded beta-pleated sheet. These are held by hydrogen bonds.
Okay, primary sequence, secondary coil sheets.
Then, the whole polypeptide chain folds up into a specific, complex, three-dimensional globular shape called the tertiary structure. This involved interactions between the R groups, hydrogen bonds, ionic bonds, hydrophobic interactions.
So the final 3D shape.
For many proteins, yes, but some consist of 2 or more polypeptide chains interacting. That arrangement is the quaternary structure. Hemoglobin is an example.
4 levels. And that final 3D shape is essential for function.
Utterly essential. Structure dictates function.
We can group proteins based on shape too, right? Fibrous versus globular.
We can. Fibrous proteins are strand-like, extended, insoluble in water. Very stable. Think collagen, tendons, bones, keratin, hair, nails, elastin, muscle proteins like actin and myosin. Mainly structural or contractile roles.
Strong ropes and cables.
Basically. Globular proteins, on the other hand, are compact, spherical, water-soluble, and chemically active. These are the functional proteins.
Like antibodies, hormones, enzymes.
Exactly. They do the dynamic jobs in the body.
Now what happens if that complex 3D structure gets messed up?
Protein denaturation. If globular proteins are exposed to harsh conditions, like excessive heat or extreme pH.
The weak bonds holding their shape, like hydrogen bonds, break.
Right. The protein unfolds, loses its specific 3D shape, it denatures.
And loses its function.
Completely. Because the function depends on that precise shape, particularly the shape of its active site where it binds to other molecules. Denaturation is often irreversible.
Like cooking an egg white. The proteins denature and solidify.
Perfect analogy. You can't uncook it. Physiologically, this is serious. Denatured enzymes stop working. Denatured hemoglobin can't carry oxygen.
So maintaining protein structure is vital. Let's focus on enzymes since they're such important globular proteins.
Yes, the biological catalysts we mentioned. They speed up reactions incredibly, making life possible at body temperature. Chemical traffic cops.
How exactly do they speed things up?
They work by lowering the activation energy of a reaction. That's the energy needed to get the reaction started.
Like giving it an easier hill to climb.
Exactly. By lowering that barrier, the reaction can proceed much faster, even without high temperatures that would denature proteins.
Clever. What's the mechanism? How do they interact with the molecules?
It's often described as a lock-and-key mechanism, though it's more dynamic, like a glove fitting a hand. The reactant molecules called substrates.
Bind to a specific region on the enzyme called the active site.
Right. The active site has a shape that's complementary to the substrate. Binding forms an enzyme-substrate complex. The enzyme might even change shape slightly to grip the substrate better induced fit.
Okay, substrate binds to active site. Then what?
While bound, the enzyme promotes the internal rearrangements needed to convert the substrate into product.
It facilitates the reaction.
Yes. Then, the enzyme releases the product. And the crucial part, the enzyme itself is unchanged.
So it can go back and bind another substrate molecule.
Immediately, ready for another round. That's why cells only need tiny amounts of each enzyme. They're highly efficient and reusable.
And they're often named for the reaction they catalyze ending in ase, like hydrolases, oxidases.
Typically, yes. The suffix ase usually indicates an enzyme.
Okay, proteins are amazing. Final class of organics, nucleic acids.
The information molecules. DNA and RNA, the largest molecules in the body.
Also polymers. What are their monomers?
Nucleotides. Each nucleotide has 3 parts. A nitrogen-containing base, a 5-carbon pentose sugar, and a phosphate group.
Base, sugar, phosphate. Got it. Let's compare DNA and RNA.
Okay. DNA, deoxyribonucleic acid, is usually found in the cell nucleus. It's typically double-stranded, forming that famous double helix shape.
And its sugar is deoxyribose. Bases are adenine A, guanine G, cytosine C, and thymine T. A pairs with T, G pairs with C.
Correct. DNA has 2 absolutely fundamental roles. 1, it replicates itself before cell division, ensuring genetic continuity. 2, it provides the master blueprint, the instructions for building every protein in your body.
It determines everything about us, essentially.
Pretty much your genetic code.
Okay. What about RNA, ribonucleic acid?
RNA is mostly outside the nucleus. It's usually single-stranded, its sugar is ribose, and its bases are A, G, C, and uracil, U instead of thymine. U pairs with A.
Single-stranded, ribose sugar, uracil instead of thymine. Its role.
RNA acts as the messenger and interpreter for DNA. It carries DNA's protein-building instructions out to the cytoplasm and helps assemble the proteins. Think of it as DNA's molecular slave. Different types exist. Messenger RNA, mRNA, ribosomal RNA, rRNA, transfer RNA, tRNA, each with a specific job in protein synthesis.
DNA holds the library. RNA checks out the books and builds things based on them.
Nice analogy. That captures it well.
Okay, we've covered the major molecules. Now let's tie it back to energy. So what does this all mean? We break down glucose, a carb, for energy, but how do cells use that energy?
Right. Cells can't just burn glucose directly for most tasks. The energy released from breaking down glucose and other fuels needs to be captured in a more convenient, usable form.
And that form is ATP, adenosine triphosphate.
The universal energy currency of the cell.
What exactly is ATP, structurally?
It's actually an adenine-containing RNA nucleotide, but with 2 extra phosphate groups attached. So adenine base, ribose sugar, and 3 phosphate groups.
Adenosine triphosphate,
Yes.
3-phosphate.
And those phosphate bonds, especially the last 2, are high-energy bonds. Think of the triphosphate tail as a tightly coiled spring storing a potential energy.
Why high energy?
Because the 3 phosphate groups are all negatively charged and they repel each other. Packing them together takes energy, and breaking them apart releases that energy. It's an unstable arrangement ready to pop.
So how does the cell tap into that energy?
By breaking the terminal last phosphate bond through hydrolysis, ATP plus H2O ADP, adenosine diphosphate, plus pi, inorganic phosphate, plus energy.
Breaking off one phosphate releases usable energy.
Exactly. And that released energy is what directly powers cellular work. Then the cell uses energy from fuel breakdown, like glucose oxidation, to stick that phosphate back onto ADP, regenerating ATP.
So it's a constant cycle. ATP, ADP plus pi plus energy, and then ADP plus pi plus energy ATP.
The ATP-ADP cycle. It happens constantly, billions of times per second in your cells. ATP is made, used, remade. It's not really for long-term energy storage like glycogen or fat. It's for immediate energy transfer.
And what kind of cellular work does ATP power?
Pretty much everything. Transport work, like pumping ions across cell membranes against their concentration gradient. Mechanical work, like muscle contraction or moving cilia. And chemical work, driving those endergonic reactions we talked about, synthesizing polymers like proteins and nucleic acids.
So without a constant supply of ATP.
Cellular work grinds to a halt. Life ceases. It's that fundamental.
Wow. Okay, that brings it all together, from atoms and bonds to complex molecules and the energy that drives it all.
It's quite a journey, isn't it?
It really is. So there you have it, a deep dive into the chemistry that underlies, well, everything in the human body. Understanding this foundation, from the smallest electron interactions to the giant molecules like DNA and proteins, and how ATP fuels it all, it just gives you such a deeper appreciation for how anatomy and physiology actually work. It's the invisible engine running the whole show.
Absolutely. And thinking about the sheer complexity, the precision of these millions of simultaneous reactions, it really makes you wonder, doesn't it?
Wonder what?
Well, this raises an important question. Considering this incredible, constant chemical activity, what does that tell us about the body's own chemical engineers? The enzymes, the hormones, the intricate regulatory systems that manage to keep everything in such perfect balance, second by second, minute by minute. The control systems are just as amazing as the chemistry itself.
That is a fascinating thought to ponder, the regulation required. Incredible. Well, thank you for joining us on this deep dive into the chemistry of life. We hope this helps you get a solid footing as you explore anatomy and physiology further. Keep exploring, keep asking questions. And thank you, as always, for being part of our last-minute lecture family. We'll see you next time on The Deep Dive.